Hello and welcome to my presentation. My name is Antje Neubauer and I'm working as application specialist at the Enpresso GmbH. In this presentation, I want to talk about the patented Enbase technology because the development of all Enpresso products is based on this. You will get informed how you can take advantage by the fed batch-like technology application, and I will show you how easy and convenient the handling is. Hippocrates formulated a statement that describes our Enbase strategy in a simple way. If we could give everyone the right dose of food, not too much and not too little, we would have found the best path to health. Eat as much as you can or starving, both extremes are harmful, even to microorganisms. Our products offer a healthy diet with continuous growth-limiting glucose supply, which guarantees successful microbial growth and increased product yield. In scientific terms, the Enbase technology is a fed batch strategy. The latter is typically used today for the production of most bioprocess-derived products. Using fed batch technology in bioreactors, one reaches 10 to 100 times higher cell concentrations compared with cell densities normally obtained in shake flasks. In fed batch cultivations, a highly concentrated sugar solution is added to a bioreactor in a controlled way by variation of the pump speed. The setting is such that the glucose never accumulates in the reactor, but its concentration remains growth limiting. In such a system, we can very easily control the specific growth rate of the cells by changing the speed of the pump. And with that, we control the diet of the cells and ultimately their physiological state. Professor Peter Neubauer and his team at the University of Oulu in Finland wondered how it could be possible to use the principle of the fed batch technology in shake flasks or even in 96 microwell plates in a much more simple way to obtain the same benefits as in the bioreactor cultivation. Finally, he and his team came up with an elegant solution. Instead of an external feed container, they supplied a soluble sugar polymer to the culture, which is not a growth substrate for the cells. Additionally, they added a biocatalyst, which splits this polymer and releases the sugar monomers, which then can be eaten by the cells. By adding this enzyme in catalytic amounts, this system works exactly in the same way as a fed batch system with an external feeding pump. They call the system Enbase, which stands for Enbase enzyme-based substrate delivery. Initial tests with Escherichia coli and a 96 well plate confirmed that the idea really works. All wells were filled with the same medium, with a polymer instead of glucose, with the same amount of inoculum, but the amount of biocatalyst was varied from well to well. As I told you before, the polymer is not metabolized by the cells. In wells without biocatalyst, no growth is happening. This is the lower black curve. In wells where only little biocatalyst is added, we see a constant linear growth. In wells where more biocatalyst is added, the biomass increases higher. And this continues in this example up to a biocatalyst concentration of 3 units. However, in wells where the concentration of the biocatalyst is even higher, we see a decline. As in these cultures, glucose accumulates and is not anymore controlling the growth. This has the consequence that the culture gets oxygen limited and side metabolites such as acetate, lactate, and succinate are formed, the products of the typical mixed acid fermentation of E. coli. I want to highlight here that already in this early phase of development, ODs of 20 to 25 were obtained, which is much higher than what can normally reached in 96 well plates. For an easy handling, whole media tablets were developed, packed into bags and sterilized. One kit contains a certain number of bags suitable for 1 liter. Additionally, the biocatalyst is provided as a sterile liquid. Different product types are produced for different application areas. The first product was Enpresso B, as E. coli is used most from laboratories as a working host. Additionally, the company developed a nitrogen-free medium for scientists who want to label their recombinant proteins with 15 and for structural studies for NMR. Enpresso Y defined is designed for scientists working with Pichia pastores. NPAM 200 contains the main components of our N-based technology, the polymer, which is provided as 200 gram in powder form, and the biocatalyst reagent A. This product allows scientists to set controlled glucose delivery in fed batch mode in their own optimized medium. The protocol is adapted to a normal working day for comfortable use. Pre-culture is done in a way you normally do, by inoculation with a glycerin broth or colonies from an agar plate. For the main culture, just take such a tablet pack, open it under the sterile bench and put it in your shake flask with sterile water. You inoculate with your starter culture. Place the flask on a shaker where the tablet dissolves within a few minutes. By adding the biocatalyst separately, you have the opportunity to optimize the feed rate according to your needs. Otherwise, you only take the recommended concentration. After overnight cultivation, more reagent A and Enpresso booster are added with additional nutrients. Cell growth and production formation continue until the next morning when the cells are ready to be harvested. To date, many groups around the world have been successful in using Enpresso media for their proteins, and more than 200 scientific publications refer to N-based technology. In many cases, users of the N-based technology not only noticed a higher yield due to the higher cell density, above all, we often get a higher yield of protein per cell under the conditions of controlled metabolism compared to standard LB cultures. In most cases, the higher yield per cell is observed in studies where part of the protein is accumulated in inclusion bodies, but an active protein is also obtained. It seems that the low growth rate, combined with a controlled metabolism, favors the folding pathway, thus leading to a higher concentration of the correctly folded product. Now I would like to show you some case studies in Escherichia coli. User Circus describes in 3 articles the successful optimized expression of the very difficult to express protein, the ribonuclease inhibitor. The gene was first cloned in a plasmid library with 45 vectors. To measure the protein aggregation, a second plasmid with luciferase as reporter was inserted. Expression conditions were analyzed by luciferase measurement, soluble protein quantification, and STS page showing total soluble and insoluble protein. All of these analyses performed with cellular material derived from just one 96 microwell were possible due to higher cell density achieved through cultivations using Enpresso technology. The influence of the growth rate at the time of induction has been investigated in shake flask cultivations by induction at different time points. It's noteworthy that the best working conditions, namely to induce at growth rate of 0.22, was verified by a fed batch cultivation in a bioreactor. In addition, it shows the importance of providing screening conditions in fed batch mode. As you can see in the graph on the right, cultivation in LB medium ended with a bad result. If the strain screening activities had been performed in LB with batch mode, this strain would never have been selected. This graph shows very nicely how well parallel cultivations can be performed with the Enpresso technology, as an almost synchronous growth of the 96 well cultures is observed before the temperature shift. You may know that there is a growing interest in the production of antibodies in prokaryotic systems. In this review, the authors from New England Biolabs show a long table of results on the functional yield of various antibodies. The studies with Enpresso products were successful and even led to better results than compared to conventional media. The third example shows the production of the peptide valinomycin. In the first step, the expression of the very large protein valinomycin synthetase with two subunits of greater than 100 kilodalton each must be successful. In order to be able to produce valinomycin subsequently, the culture must still be in a metabolic state. To obtain the valinomycin synthetase in soluble form, Jacek and colleagues tested different tags and ribosome binding sites, different E. coli strains and culture media, inducer concentration, and temperature. The use of the Enpresso growth system had a major advantage here. The slow, controlled growth allowed cultivation for up to 100 hours, which not only increased the soluble form of valinomycin synthetase, but also expressed valinomycin in high concentrations. To further prolong the culture, they added the polymer at different points in time and achieved a final OD of more than 50 and an even higher valinomycin yield after 50 hours of cultivation. Now I want to tell you something about the application in yeast. Yeast generally grows slower than E. coli, so maximizing growth is particularly beneficial. Plus, in vitrogen standard protocol requires 1 or 2 pulses of methanol addition per day. The measured values shown in the graph show the effect of impulse feeding with methanol. Cells are metabolize methanol as a carbon source, and the varying presence of methanol causes stress to the cells. By applying Enpresso with a constant background feeding, a lack of carbon is avoided. Using Enpresso Y-defined means less hands-on time. The tablets must be dissolved in sterile water, no media changes are required during the cultivation process, no OD measurements are required. You can grow your culture overnight and induce the next morning. Currently, the Laboratory of Bioprocess Engineering at TU Berlin has developed the mathematical model to better understand the Enpresso systems and also to enable exponential feeding to keep the growth rate constant. The first of these models has just been published in Bioengineering 2024 by Kemmer et al. At the robot station, the Enpresso system was used where the specific growth rate could be kept constant over a longer period of time by adding the biocatalyst or polymer. You will benefit from using Enpresso products if you need to make recombinant proteins in E. coli or Pichia pastoris, as they support microbial growth. Higher final cell densities allow you to reduce your cultivation volume and so save time. It's very easy to use as the Enpresso grow systems are ready-to-use cultivation media. Reproducibility through controlled growth with Enpresso, even on a small scale, provides you with a perfect environment for screening for expression conditions to develop a bioprocess on a large scale. Thank you very much for your attention.